Good morning. In my talk, I would like to present you results of our study focused on meta-analysis of heritability and genetic correlations between resilience and efficiency traits in goats and sheep. Breeding programs in small ruminants have achieved a great deal of progress for production traits. Currently, there is a growing interest in improvement of health-related traits in order to breed more robust and resilient animals. In order to achieve this goal, it is important to get a good insight into the genetic architecture of resilience traits and their relationship with efficiency traits. Therefore, the objectives of this study were to gather estimates of genetic parameters from partners of the SMARTER project and supplement this with parameters published by other research groups based on a literature review. Subsequently, we created a database with all the genetic parameters and performed a review of available data to identify traits with a sufficient amount of information for inclusion in meta-analysis. The main goal of this project was to obtain pooled estimates of heritability and genetic correlations between the selected resilience and efficiency traits. This study was based on data collected from 13 partners of the SMARTER project. Additionally, to get a more comprehensive overview, it was supplemented with genetic parameters published by other research groups which were collected during a systematic literature review. In total, genetic parameters from over 230 papers and unpublished results from 81 sheep and 14 goat breeds were collected. As a result, a database with over 2,000 estimates of genetic parameters was created. This encompasses over 800 estimates of heritability and over 1,400 estimates of genetic correlations. The vast majority of those estimates belongs to sheep, with close to 600 estimates of heritability and over 1,100 genetic correlations. In case of dairy goats, the number of published papers is much smaller, which limited the size of the dataset to 200 heritabilities and 250 genetic correlations. Subsequently, the dataset was analyzed to select traits with a suitable number of heritability and genetic correlation estimates, together with accompanying information about each study, such as breed, age of the animals, and number of phenotypes used to estimate the genetic parameters. Only traits with a minimum of 3 estimates from 2 papers were selected for meta-analysis. In case of dairy goats and sheep, 5 efficiency traits were selected related to milk yield and milk composition. These were milk, fat and protein yield, along with fat and protein percent. In case of resilience traits, somatic cell count was selected as a proxy for mastitis and fecal egg count as a proxy for parasite resistance. In case of meat sheep, 2 efficiency traits were selected. These were body weight and growth. For resilience traits, we selected fecal egg count and parasite immunoglobulin as a proxy for gastrointestinal parasite resistance, and dugginess and fecal consistency as a proxy for fly strike. In order to account for heterogeneity of the data, hierarchical word clustering was performed to identify groups of estimates according to similar conditions in which they were obtained. Goer distance was used as a measure of similarity between data objects. It was selected because it can handle both categorical and continuous variables, which was important given the structure of our dataset. Clustering took into account variables such as breed, sample size used to estimate the genetic parameters, and age of the animals, which was based on lactation number for dairy or age at measurement for meat traits. Categorical variables such as breed were transformed into unique classes, whereas quantitative variables such as heritability, genetic correlations, or sample size were standardized with a mean of 0 and standard deviation of 1. The following 3-level meta-analysis model was fitted using an R package metaphor. The model contained 3 random effects, which were the effect of within-cluster heterogeneity, which represents heterogeneity of estimates nested within each cluster, between-cluster heterogeneity, and sampling error of individual studies. Looking at the results of meta-analysis for heritability in dairy goats and dairy sheep, traits such as milk yield, fat and protein content had a very wide range of reported values, as you can see in this figure, which represents heritability for milk yield. On the other hand, for traits such as somatic cell count, which you can see in this graph, or traits such as fat and protein yield, as well as fecal egg count, the estimates were quite close to each other. Pooled estimates of heritability for production traits in dairy goats were between 0.27 and 0.48, whereas resilience traits had heritabilities of 0.07 for fecal egg count and 0.21 for somatic cell count. Similar results were obtained for dairy sheep, but with slightly lower estimates for production traits. It is also worth noting that the number of observations reported in the literature was much smaller for resilience traits. Meta-analysis of genetic correlations between resilience and efficiency traits in dairy goats was based on a very small number of observations, as the number of published papers for these traits is very limited. Additionally, the range of reported values is often very wide, as is the case with fecal egg count and milk yield, where it ranged from minus 0.21 up to 0.63. As a result, most of the pooled estimates, except of the correlation between somatic cell count and fat percent, were not significantly different from zero. Much more estimates were available for genetic correlations between resilience and efficiency in dairy sheep. Similarly to the analysis of heritability, also in the case of correlations, as you can see in this graph, which represents a genetic correlation between somatic cell count and milk yield, the range of reported values was very wide. In case of genetic correlation between somatic cell count and milk production traits in dairy sheep, the correlations were unfavorable for somatic cell count and fat percent, as well as protein percent. For the remaining traits, the correlations had large standard errors and did not differ significantly from zero. In meat sheep, also a wide range of genetic correlations has been reported in the literature, as can be seen in this graph for body weight and fecal egg count. In this case, correlations ranged from close to minus 0.9 up to 0.34. As a result, the pooled estimate of genetic correlation was close to zero. This situation, with a wide range of estimates reported in the literature, leading to pooled estimates which did not differ from zero, was quite common. Only the pooled estimates of genetic correlation between growth and fecal egg count and growth with dagginess were significant and negative. To conclude, we can say that based on the performed analysis, based on the analyzed data, we could see that the number of traits as well as the number of studies which have reported genetic correlations between resilience and efficiency traits in sheep and particularly in dairy goats is very small. This indicates the need for more research in this area in order to obtain a better insight into the relationship between these traits and to properly include them in breeding programs. Correlations between the analyzed traits were generally unfavorable, which further strengthens the need for estimation of accurate parameters to model and predict the impact of selection on resilience and efficiency traits. Further studies will also allow us to gain a better understanding of the mechanisms underlying the relationship between these traits. I would like to thank you for your attention and acknowledge all partners of the SMARTER project who contributed data to this analysis.